Hi guys, and welcome back to week 6. This week we're going to be talking about developmental networks. This is an example of the developmental network that we're going to be looking at this week. We can see it here again in the left. So here the end product is the formation or expression of spikes. Our arrows represent activation and our bars represent repression. So we want to know who will and who will not produce spikes. So our wild type, which is our regular or our normal expression, meaning no mutants, our wild type, S will activate P to inhibit I will then inhibit K, and K will then activate E, which is our end product. E will activate and express the trait of spikes. For I and E, I will not be able to produce spikes because it inhibits, but since E is the end product, spikes will be expressed. For S and K will both be able to produce spikes as they are activators. P will not be able to produce spikes because it is an inhibitor, and I will also not be able to produce spikes as it is an inhibitor. So why don't all cells produce spikes? So this can be explained by a type of regulation called alternative splicing. This happens during post-transcriptional regulation. And this is a single gene can contain numerous exons and introns, and the exons can be spliced together in different ways. For example, if a gene contains 10 exons, one version of the mRNA transcribed from that gene might contain exons 1 through 9. Another version of the mRNA might contain exons 1 through 8 and exons 10. This is called alternative splicing and can produce different forms of a protein from the same gene. We can see a picture of this below to help us understand what that explanation means. These are my references. Thank you for listening.